Hello, everybody. When Watson and Crick first published their proposed structure of DNA, one of the things they said in the paper is that they realized that the structure itself suggested a way that DNA could be replicated, which is what we are going to talk about today, the replication of DNA. Turn off the video and see, since you now know what the structure of DNA is like, if you can figure out how it might be replicated based on that structure. In addition, in today's talk, we are going to learn for the first time what enzymes actually do. And since enzymes are gene products, they're going to give us an understanding of how our genetics works. So let's get started. I want you to recall that DNA is a long linear molecule, and on this long linear molecule, genes are located from place to place. And we call these long segments of DNA chromosomes. And in eukaryotes, they're a complex of DNA and proteins. But we're not going to discuss how the proteins function in the structure of DNA. That will be left for another course. Each gene occurs at a particular location on a chromosome, and it consists simply of a sequence of the bases A, C, G, and T. And that, in fact, is what the entire molecule of DNA consists of. But the coding regions are just a sequence of A, C, G, and T. So one of the things we're going to want to learn eventually is how do those A's, G's, C's, and T's carry information? And we'll get onto that soon. There are many genes on each chromosome, and in humans, perhaps as many as 2,000 on our largest chromosomes. Each individual gets a complete set of chromosomes from its mother and a complete set from its father. So this means each individual gets 2 copies of each gene, one from its mother and one from its father. Now I want to go on and talk about something that may seem unrelated, but that is growth. What causes growth of an organism? Well, think about it. You began as a single-celled organism, a fertilized egg, a zygote, and now you are 30 trillion cells as you get into your adult body. How did that happen? Well, it happened by cell division, cells making copies of themselves. Cell number increases through cell division, so we can say cells multiply by division, and this is why cells are really mathematically confused. If you think about it, for cell division to occur, the DNA has to be replicated beforehand. So how does this happen? And one clue of how this happened is if you think about this, and this was the realization that Watson and Crick had. If you pull the strands apart, can you tell what the paired bases should be? Well, of course you can. You know that opposite a G, there's going to be a C, opposite a T, there's going to be an A, opposite a C, there's a G, etc. That's what we call complementary base pairing. Since the 2 strands of DNA are complementary, each strand acts as a template for building a new strand in DNA replication. What's going to happen when DNA has to be replicated is the parent DNA molecule is going to unwind, and 2 new daughter strands are going to be built based on the base pairing rules. How would this piece of DNA be replicated? Well, you already know the answer. It would be very easy for you to fill in the missing base pair. In other words, this here would be a C, this would be an A, this would be a G, this would be a T, etc. You can see it's right down here. And the opposite side would be paired with a G, a T, a C, and an A. Here's an overview of the replication of DNA. DNA is pulled apart, that's the first step, and then in the second step, complementary base pairs are matched, and the phosphate sugar backbone is made, and then the hydrogen bonding occurs between the new base pairs, and you have a strand of DNA. And the new strand consists of 1/2 of it is an old strand, designated in dark blue, and 1/2 of it is a new strand in light blue. As a result, we call this replication semi-conservative. In other words, each daughter molecule has 1 old strand and 1 newly made strand. It could have been otherwise, and there were several hypotheses, and they looked like this. It could have been conservative replication, where a new strand is made from the old strand, or it could have been dispersive, where there's a mixture of new and old in each strand. But what the evidence showed ultimately was that it was semi-conservative replication. I'll leave you to go back and look at this on your own time. Now, DNA replication is carried out by enzymes. So let's remind ourselves, what are enzymes? They're proteins that catalyze chemical reactions. And what's a catalyst? A catalyst is a molecule that increases the rate of the chemical reaction, but is not changed by the reaction itself, and so can keep causing the reaction to happen. So, in other words, enzymes are proteins that facilitate chemical reactions in the cell. In DNA replication, there are 5 enzymes that you're going to need to know about, and here are their names. They are helicase, and it unwinds the DNA. DNA polymerase, there are 2 different kinds, and they make the new strand of DNA. There's primase, and it gets everything started. And finally, there's ligase, and that ties up the loose ends. And we're going to see how this works in just a second. One thing for you to note here is that the suffix ase signifies that the protein is an enzyme. DNA polymerase has to build the new strand. However, it can only work by attaching new nucleotides to an existing 3' end. You should know that this up here is the 5' end, 5' here. And this down here is the 3' end. And on the other strand, this is a 5 and this is a 3. The 2 chains run in opposite directions, as we already know and as I've just shown you. As I said, DNA polymerase can only add nucleotides to the existing 3' end. Let's take a look at what that means. Here we have the template strand. Here we have the new strand. Here is the 5' end of the new strand. That means that the DNA polymerase can only add to this end. Remember that because the DNA strand is antiparallel, the 5' end here is matched with a 3' end in the template strand. So DNA can only elongate in that direction because of the limitations of DNA polymerase. There are numerous confusing consequences of these limitations of DNA polymerase. The first is that the new strand can only grow in that one direction, which we've already talked about. The second is because the 2 strands are oriented in opposite directions, the direction of copying is in the opposite direction on each strand. When a DNA molecule is pulled apart, one of the strands is replicated in one direction, the other is replicated in the opposite direction. And finally, since DNA polymerase requires an existing 3' end, you may be asking yourself, how does the first nucleotide of the new strand get put into place? And that's a really good question. If that had occurred to you already, pat yourself on the back. A really excellent question. One of the template strands, the new DNA, is synthesized continuously because there's always a 3' end available. I'm going to show you a picture of this and go over this again. This is called the leading strand. The point of the leading strand is there is continuous copying. Along the other strand, which is called the lagging strand, the DNA polymerase is constantly running into a 5' end of an already copied piece of DNA. And so a gap is left, and I'm going to show you that in a second. The lagging strand, therefore, is synthesized as a series of fragments, and they're called Okazaki fragments after Dr. Okazaki, who discovered them. So let's take a look at this in pictures. Here we have the leading strand, and here we have the lagging strand. Let's look at this. Here's the 5' end of the parent DNA here. That means this over here has to be a 3' end. This is the 3' end of this parent DNA, so that means that has to be a 5' end. If this is a 3' end up here, then this has to be a 5' end, and then this right here is a 3' end, okay? So that means that this molecule here, which is DNA polymerase, is able to just keep copying as the DNA is unwound here. As the DNA is unwound, this just keeps copying. On this end, however, there's new DNA opened up. And so when DNA polymerase attaches here and starts working this way, eventually it's going to run into an already produced piece. So this side gets put together as a set of fragments, the so-called Okazaki fragments. And you can see once again on the leading strand there is continuous copying into the place where the DNA is being unwound. That's called the replication fork. But on the other strand going away from the replication fork, the DNA is constantly running into an already created piece of DNA. We're going to review this again in a little while. But that's one of the first confusing aspects of it, is the leading strand gets copied differently than the lagging strand. Let's now do DNA replication in its full glory. First, the enzyme helicase separates and unwinds the parent DNA and seems to play a central role in organizing all of replication, as you will see in the video at the end of this talk. Second, the enzyme primase puts down a small piece of RNA primer to begin the process of making a new strand. Now, why does it do this? Because DNA polymerase 3 can only work with an existing 3' end. And so the primer gives a 3' end that the DNA polymerase is able to work with. DNA polymerase 3 is the one that builds the new strand from the primer. And yes, there are many different DNA polymerases. We're only going to learn about 2 of them. And it builds the new strand until it runs into the next piece of RNA primer. And it can't deal with that, and so it stops working and detaches itself. That's where DNA polymerase 1 comes in. It attaches itself and is able to remove the RNA primer and replace it with DNA, but when it eventually bumps into a piece of DNA, it cannot close the final gap. All right, we need a separate enzyme called ligase, and ligase is able to close the gap between the pieces of newly made DNA. In a second, we're going to see this in pictures, but I just want to point out that if you had designed this process, if you had been a designer for life, you probably would have made a single enzyme that can do all these things. And this ridiculous complication and limitation of each of these enzymes is an indication that life evolved and wasn't designed. So here now are the 5 steps in all their glory. So we're going to start with the lagging strand. Here is helicase, and you should have this diagram, so you should label it as we go. Here is helicase, and then primase comes along, and you can see the red nucleotides being put down are RNA nucleotides. Once there is a primer laid down, DNA polymerase 3 can attach and start to build the new DNA represented by light blue, and it will continue working until it bumps into the next primer. At that point, it detaches, having built, in this case, a small section of new DNA. DNA polymerase comes along, and it now removes the RNA and builds the rest of the DNA, but it cannot close the final gap. And there you see now the final gap in this next section. That is left to the enzyme DNA ligase. So there are the 5 enzymes in action: helicase, primase, DNA polymerase 3, DNA polymerase 1, DNA ligase. Notice that we did this for the lagging strand, and you can see the Okazaki fragments here. Fragment 1, fragment 2, fragment 3, fragment 4. They're all nicely labeled. On the leading strand, notice in this picture, the primer was laid back way back here out of the picture. And since then, DNA polymerase has just been able to keep copying into the opening of the DNA. And it won't be until it comes up to another one of these bubbles of replication that it will run into a piece of primer and therefore go through the DNA polymerase 1, DNA ligase situation at that point. There's the leading strand copied continuously, while the lagging strand is copied in segments. In class, we will watch this animation. I think you'll enjoy it. And that is the end of today's talk. What I hope you now understand is how DNA is replicated semi-conservatively, how each of the strands of the parent DNA serves as a template for making a new strand, how there are 5 major enzymes involved in the process of replication: helicase, which unwinds; primase, which puts down an RNA primer so that DNA polymerase 3 can start building the new strand; DNA polymerase 1, which removes the RNA that DNA polymerase 3 can't and builds the rest of the strand until it runs into the next piece of newly copied DNA, at which point it detaches, and finally the final gap is closed by ligase. So, until next time, go outside, take a look at nature, ask biological questions, and bring them back to class. And I'll see you soon.